Antibodies, and that brings us to the next topic in that line, that is antigenic determinants of immunoglobulin. Now we know antibodies are gamma globulins and they are proteins, but they are glycoproteins and they can themselves act as antigen or immunogens. But not entire antibody molecule is immunogenic. That means they can generate immune response, but only some portion which can generate antibody response in other individuals if they are introduced in them. And such portions are known as antigenic determinants of immunoglobulin. So consider these as sites which produces antibodies in other people if your antibodies are injected into them. So based on location of these antigenic determinants, they are divided into three types. The first type that I'm going to discuss is isotype. Now, isotype, the classical example, if I can give you, is your 5 classes of immunoglobulin. IgG, IgA, IgM, IgD, and IgE are basically isotypes. IgG 1, 2, 3, 4 are even a further example of isotypes. So they vary from each other. They are different from each other. How they are different? Because their heavy chain is different. And the constant region of heavy chain shows variation among each molecule. So the change that you can see among them is present in the constant region of heavy chain. So they are different among species, but they are same in all members of one species. So they show interspecies variation. So that is about isotype. Let us understand the next type, that is allotypic determinants. So these allotypic determinants, you can understand that they are present in isotype genes in obviously constant regions, here also in H as well as L chain, though I am showing it in H here in but if you can closely see the diagram, you will also appreciate that L chain is also showing variation, while in above the L chain was remaining the same. So in isotype there is no change in L chain, but here constant region of both H and L chain are showing the difference. And here the antibody molecules are different among species, and they're only found in some member of one species. So it shows intra-species group variation. They're only found in some individuals of one species, but they have variation or they are different in each species. So here gamma heavy chain has 25 GM types, alpha heavy chain has similarly various types, AM systems is there, kappa light chain system is there, and it has 3 KM allotypes. So the individual allotypic determinants displayed by an antibody determines its allotype. So that is about allotypic determinants. And lastly, we have idiotypes. So in idiotypes, you can see a unique amino acid sequence present in paratop region of one member of a species. And such antigenic determinants are known as idiotypes, and some of these idiotypes constitutes this idiotype. So they are different among species, as well as they are different in every individual member of a species. So they are same in identical twins though. So intraspecies individual variation is there, and here the change is in both light and heavy chain, but in variable region or antigen binding region. And sum total of idiotypes of an immunoglobulin molecule constitute idiotypes. So this is a very important topic and you can be tested on various concepts, especially the thing that you should know that how they are different, what are the examples of them. And idiotype allows us to have the possibilities of 10 raised to 8 different antibody molecules against different antigen. So the large capacity to neutralize the antigen is possible with these idiotypic determinants. So that's all for antigenic determinants of immunoglobulins. I hope you have understood this clearly. If you have any doubts, post your doubts in comments and I will try to answer them uh, as best I can. Thank you for listening to me. Next session, we will talk about monoclonal antibodies.